Hello biology students, and welcome to this video where we're going to be discussing our new unit, which is a new unit all about genetics. So genetics as a unit is divided into two parts. The first part is Mendelian genetics, which is going to be looking at all of the genetic rules that this man named Gregor Mendel discovered. And then the second half of our unit will be discussing non-Mendelian genetics. So in this first part, these are the topics we're going to hit. And in this video specifically, we're going to look at Gregor Mendel, who he was and what he did, and also some basic genetics vocabulary. So before we start talking about genetics, it would be good to have a definition of what genetics was. So genetics is the study of heredity. And if you don't know what that word heredity means, heredity is the idea that you can pass on a characteristic or a trait from a parent to an offspring. So really, in genetics, we're looking at how traits get passed on from one generation to the next. A geneticist, a modern-day geneticist, studies what are called genes, and we've seen that word before. Remember, a gene is a stretch of DNA that codes for a protein. And remember, those proteins and their functions are what ultimately give us our traits, our physical traits. So if we're looking at how traits are passed on from one generation to the next, really we're looking at how genes or DNA are passed on from one generation to the next. So the first person who really discovered anything about genetics was this guy named Gregor Mendel. And Gregor Mendel was a monk who lived in the mid-1800s, and he basically raised pea plants, like the green peas that you eat, in the garden in his monastery. And by kind of observing these pea plants and doing experiments with these pea plants, he figured out the basic laws that we use in all genetics today. So because he figured this all out, we call him the father of genetics. He was kind of the first person to kind of learn about genetics and genetics principles. Now, something really impressive about his work is he figured out how DNA and traits are passed on from one generation to the next before anyone even knew what DNA was. That's quite impressive. So there are a lot of reasons he used pea plants. The first is that they're really easy to grow and manipulate. Pea plants are plants, obviously, so they can't walk away from you. You can keep them in certain pots, keep them organized. They're easy to work with. They mature very quickly, which means you go from a young pea plant to an adult pea plant in a very short period of time, which means you can study several generations of pea plants in a short period of time. Now, if we were studying humans, it takes us about 15 years to go from an infant to reproductive age, the age at which we can have our children ourselves. So that would be kind of hard to study how traits are passed down from one generation to the next if we use something that lived long like humans did. And pea plants also show contrasting traits, which means it's very obvious what trait they have. They're tall or they're short. They have purple flowers or they have white flowers. There's no in between. And this word trait we're going to define as a characteristic, or basically a heritable feature, a feature that's dictated by DNA that varies among individuals. So an example is flower color. And Mendel, in particular, studied 7 characteristics, or 7 traits, that he used to figure out his genetic information that he discovered. So a little bit about pea plants themselves, or plants in general. So we look at this picture. This is a picture of a pea plant. And in order to fertilize a pea plant, what you do is in the flower itself, the flower is the reproductive organ. And most plants, or most, not all, but most plants have both female and male reproductive parts. So the carpel, this part in green right here, that's the female reproductive organ. Then the stamens with the little yellow tips, those are the male reproductive organs. And pollen, which is the yellow stuff at the end of those stamens, that's plant sperm. Now you can take pollen or plant sperm from one plant and you can touch it to the end of this carpel, and then the little pollen cells or pollen granules will travel down into the carpel and fertilize the plant egg. When that happens, you get a plant zygote and plant embryos, which mature into seeds, which in the case of the pea plant are peas. So when you see a pea, that is actually a little fertilized plant embryo or a seed that you can then plant and get plant offspring. So Mendel used this basic method to study how different traits, the ones listed right here, are passed down from one generation to the next. So he looked at these 7 traits that are listed on the right. So Mendel did two things in his crosses. So because plants have both female and male reproductive organs, you can self-pollinate or self-fertilize, which means you can use the sperm or pollen from a plant to fertilize itself, to fertilize the egg that exists in that plant. This is still sexual reproduction because we still have a genetically unique sperm that has a random subset of half the amount of DNA from that plant and a genetically unique egg that are combining. So it's still sexual reproduction, it's just from the same parent. Okay. Now, some plants, when you self-fertilize them, will always make offspring that look just like them, okay? So we call this true breeding. So, for example, if I took a true breeding short plant, and again, not all plants are true breeding, just certain plants. If I took a true breeding short plant and I pollinated itself, I would always get short plants out in the end as offspring. That's what true breeding means. If you cross it with itself, you get plants that have the same trait. So that's a vocabulary word you're going to see here and there while we're working with some genetic problems. The other thing you can do with plants is this thing called cross-pollination or cross-fertilization. And that's where you take the pollen or the sperm cells from one plant, from one pea plant, and you fertilize the egg from a different pea plant. So this is kind of the typical sexual reproduction that we're familiar with, right? Two parents making an offspring or making one seed. Okay, so let's talk a little bit about the experiments that Mendel performed. So he had some very, very famous experiments that every biology student, in the country knows about. So the first thing Mendel did was he cross-pollinated, aka he had two parents, two true breeding plants. Now remember, true breeding means if you cross it with itself, it will always produce offspring that looks like it. So these two plants he called his parental generation, the parents. We're going to abbreviate that the P generation. So whenever you see P generation, we're referring to the parents of a cross. And what he did is he took a true breeding short plant, so a short plant that when you cross it with itself, it always had short offspring, and he crossed that true breeding short plant with a true breeding tall plant. Remember, true breeding tall means if you crossed that tall plant with itself, the offspring would always be tall. And he was curious what was going to be the outcome, what was going to happen. So when he did this, he took his true breeding tall plant and he crossed it. So he took some of the pollen or the sperm from this flower and he put it on the egg of this plant, right? So he crossed that plant with a true breeding short. Now this kind of cross, when we're taking two parents and we're only tracking or only really caring about one characteristic, or in this case plant height, we call this a monohybrid cross. Okay, so this is an example of a monohybrid cross. Now Mendel was curious, if you have a plant that always makes tall offspring and you cross it with a plant that always makes short offspring, he was curious what was going to happen. So he crossed these plants together, and to his kind of shock or discovery, all of the plants, all of the offspring when you cross tall with short, all of them were tall. And he was curious, well, what happened to the shortness, right? You had this true breeding tall plant, you crossed it with a true breeding short plant, and all the offspring were tall. Where did the short go? So another vocabulary word for you, these tall plants, the offspring that we got, we call this the F1 generation. So you can think of it as the F1 generation are the offspring, the children of the parental generation. Okay, if you know Latin, F1 stands for filial one. Filial means son, like sons and daughters. So these are the offspring of the parental generation. So now, so this picture kind of just shows you what Mendel did again. Took his tall parent, his true breeding tall parent, crossed it with the true breeding short parent, and then he got these offspring, and they were all tall. This short gene from this parent was gone, and this kind of puzzled him. So the next thing Mendel decided to do is he took this plant right here and this plant right here, the two offspring, and he decided to cross those two plants together. He was curious if he fertilized this plant with this plant, well, what would happen? So this next cross is that Mendel allowed his two F1 plants, his two offspring plants, to self-pollinate, those two talls, and then he got some more offspring from there. So these offspring are called the F2. So the F2 are the offspring or the children of the F1. And he saw here that 3/4 of the time, 75% of the time, the F2 offspring were tall. However, 1/4 of the time, those offspring were short. So even though the shortness gene had gone away in the previous generation, it's now come back. So the shortness gene has reappeared. So this really puzzled Mendel, and he had to think about it for quite a while to try to figure out what was happening. So kind of to summarize Mendel's entire experiment, he took a true breeding tall plant, crossed it with a true breeding short plant. That's his parental generation. When he did that cross, all of his offspring in the F1 were all tall. But then when he crossed this tall plant and this tall plant together, he got a bunch of offspring, which we called the F2, and 3/4 of the time, 75% of the time, he got a tall plant, but 1/4 of the time, he got a short plant. So the shortness has reappeared. It's skipped a generation. So he thought about this for a while, and he was kind of puzzled by it. But eventually, he made a hypothesis. And he hypothesized that plant height, and really any other gene, has what we call two alleles, or two versions of a gene. An allele we define as a version of a gene. So if we're looking at the height gene, the two versions, or the two alleles for that gene, would be the tall version and the short version. And he hypothesized that in his parent generation, in his true breeding tall plant, he assumed that those tall parents passed on a tall allele, a tall version, to their offspring, and he assumed the short true breeding plants passed on a short allele to their offspring. So then he figured, hey, if one parent gave their offspring a tall allele and one parent gave their offspring a short allele, that must mean those F1s have one of each. So the F1s have both the tall and the short version. However, even though they had one of each, one tall and one short, they were tall in appearance, which means that the tall was winning out. It was winning out over the short, okay? And the fact that the tall won out over the short, we call that the tall to be the dominant allele. The dominant things are the winners, right? So the tall wins out, it's dominant over the short. And we call this short allele the recessive. If you're recessive, that means you get hidden if a tall allele is present. So basically, the tall allele is stronger than the short one, and it masks or hides that short allele that's in the F1 plants. So Mendel came up with what he called his law of dominance. And the law of dominance states that some alleles are dominant over others, just like tall was dominant over short. So the way a dominant allele works is you only need one allele, one copy of that gene to show that trait. Okay, so typically we use letters to represent our alleles, and if we're talking about a dominant allele, we're going to use a capital letter. So if we decide to use T as our letter for the height gene, the capital T would represent the dominant allele, the tall allele. So remember, everyone has two copies of each gene. So if you had two capital T alleles, if that was your two versions of your height gene, you as a pea plant would be tall because you only need one copy of the dominant allele to show that trait, to show that height. And if you had one big allele for the dominant and you had one little allele, which was short, because the dominant wins out over the other one, a plant that had these two versions of their genes, because we know that we each get one from mom and one from dad, you would still be tall because that capital T is winning out over that little t. So the other allele, the little t, we call the recessive allele. And in order to show a recessive characteristic, you need two copies of the recessive allele. Or in other words, you need 0 copies of the dominant, because if you have even one copy of that dominant allele, it's going to mask or it's going to overpower the recessive. So the recessive allele gets hidden when it's paired with the dominant allele, and therefore the only way you'll show the short or the recessive trait is if you have two recessive alleles. You would have to have little t as the two versions of the genes that you got from your parents in order to be short. Okay, so finally we have two more vocabulary words I want to look at, and those words are genotype and phenotype. So genotype is a word that we use to describe the alleles you have. So when we're talking about does someone have the dominant allele, the recessive allele, one of each, two of the same, we use the word genotype. So like a potential genotype for a tall plant would be capital T or capital T little t. A potential genotype for a short plant would be little t, because those are the genes, those are the alleles that plant would have. Your genotype can also be described in one of three ways. You can be heterozygous. Hetero means different. That word hetero, that root word means different. Zygos means zygote. So a heterozygous individual would have two different alleles because hetero means different. So someone who was capital T little t, they would have a heterozygous genotype. Homozygous, homo means same, that prefix. So if someone was homozygous, they would have two of the same allele. And homozygous comes in two flavors. You can be homozygous dominant, which means you have two of the same dominant allele, capital T, or homozygous recessive, which means you have two of the same recessive allele, or little t, little t. Okay, so then finally, the last vocabulary word we're going to introduce, which is the opposite of genotype, is phenotype. So phenotype is what trait or what characteristic you actually have. Like what do you actually look like? Are you tall or are you short? Okay, so if your genotype was capital T, your phenotype would be tall. And if your genotype was little t, your phenotype would be short. So a good memory trick is genotype is the genes, aka your capital T's and little t's, where phenotype is the physical feature, lots of F sounds, right, that you see. So phenotype is what you see, genotype is your genes. So then finally, just a reminder, like in case you're wondering, why do pea plants have two alleles? Like why do they have two letters? Like why do they have two copies of each gene? Well, remember, one came from your mom and one came from your dad. So you have two copies of each gene. Remember, we call those two chromosomes a homologous pair, where if this purple and pink part are genes, perhaps your mom's chromosome gave you the capital T and your dad's chromosome gave you the little t, right? So that's why you have two copies, a little throwback to our previous unit with meiosis. So that's it for this video, folks, and I'll see you in class.